So, hi everyone. Thank you for having me. I should start off by saying that today I'm representing Jalmar Theo, who is a PhD student in our lab, and he did all of this work, but his wife had a baby just last week, so he couldn't be here. So I'm going to talk about genetic influences on cortical functional connectivity in adolescents. And in the few next slides, I will try to summarize some of the work that has been done in the field. Doesn't move. Oh, it's the other one. So this is my poor summary of what has been done in functional connectivity studies in adolescents. So if I would have to summarize this in one sentence, the brain shifts from more integrating within networks and segregation between networks. So obviously that doesn't do justice to the vast literature, so please read these reviews if you like. But I would like to emphasize that longitudinal studies into this subject are very rare. I know of 11 studies that did longitudinal functional imaging in adolescents, and most of them are focused on specific connections only. No, wrong. There you go. So another ingredient of this study is heritability. And again, the literature of heritability and functional connectivity is pretty large, but there are only a few done in adolescents. So these three studies above, one of which is our own, all show moderate heritability of functional connectivity measures in childhood and adolescence. And the other study I would like to highlight is a longitudinal twin study which was done in infants, and they looked at heritability of functional connectivity between birth and 2 years of age, and their heritability seemed to increase. And the reason I highlight this study is because the design and the type of analysis that they did is very similar to ours. So this is our cohort. It's the BrainScale cohort, with a collaborative effort between the University Medical Center in Utrecht and the Free University in Amsterdam. And what we did there is to invite 9-year-old twins and their older brother or sister to come in, and we assessed them on a number of domains, including resting state fMRI. And then we repeatedly asked them 2 times to come back for follow-up assessments. And we have resting state available at age 13 on average and age 18. So there is the unique feature of this cohort is that it is 1, longitudinal, and 2, that it is a genetically informative sample. Because if you look at related data, you can get an estimate of heritability at each time point. But what we can do, since it is longitudinal, we can also look at how the genetic influences change across adolescence. So maybe there are different genes into play at the end of adolescence at 18 compared to genes that are into play at age 13. So this is a very brief summary of what we did with the data. I'm happy to talk to you about it later. But we have about 9 minutes of resting state data acquired. And then we did a multitude of methods to try to get rid of motion, which is obviously a problem in this age range. And we ended up with losing about 20% of our data. And so the final sample is 315 subjects. And so if you wonder why we have more data at the third wave, the second follow-up, than at the second, I guess that would be because age 13 apparently is the age in the Netherlands where most children get dental braces. So then we proceeded and computed resting state networks. And we used this atlas you see here on the right, which is based on the Human Connectome project data. And we computed within resting state networks connectivity and between resting state connectivity. And in the lower diagonal, you see what happens at age 13. And in the upper diagonal, you see what happens at age 18. And I'd like to stress that this is what happens on a group level. So these are the means. And it looks pretty symmetric, so that would suggest some kind of stability over time. But since we are interested in individual data, because that's what you need if you want to look at the genetics of such a phenotype, we decided to look into our data in a little bit more detail. And so what we did is to take our resting state signal, 900 frames, and then split it up into two halves. And then repeated all this analysis and computed functional connectivity within networks and between networks. So what you see on the right is organized in the same manner as the picture you saw before. So on the lower diagonal, you see what happens at age 13, and on the upper diagonal, you see what happens at age 18. But instead of functional connectivity, we now have mapped the within-session reliability of the functional connectivity. And it is about 0.35. So it's reasonable, but it is not that great. But we decided to make use of the fact that we now had these two half scores, or so we call them, because what we would like to do is to extract something that is common to these both half scores, and that we call the reliable component of the functional connectivity. And then we applied this model. And I realize it can be a bit intimidating, but I will walk you through it. But it is important to know, if you do an analysis like this, it is not so much about the means, but it is all about separating the variance into meaningful components. So that's what we will do. And over here, these are the actual data in the squares. So we have 4 half scores, 2 at the first wave and then 2 at the second wave. And this is where the actual data goes in this model. And then we separate it in something that is common to the first time point and common to the second time point. That's the red box above. So these F1 and F2 are factors that measure this so-called reliable component. And at the bottom, there's the error variance. And that could be anything from true measurement error, but also things that happen to be very dynamical, because that would not be captured by these two half scores in the same manner. And then here on top, we put a so-called traditional twin model. So on these reliable factors, we try to separate those into additive genetic effects, common environmental effects, and unique environmental effects. And then on the left, these 3 circles represent the things, again, that is shared between these two factors. So the reliable factor at age 13 and the reliable factor at age 18. And on the right, there are 3 separate circles, A2, C2, and E2. And if you would find those to contribute significantly, you can interpret that as being separate genetic factors that act on age 18, but not on age 13. So you could interpret that as the new genetic factors that come into play, even it's a big stretch, but you could also interpret that as maybe increased gene expression of certain genes. So now for the results. These are the mean effects of age and sex on functional connectivity. So in each of these balls, each of these balls is a resting state network, and then within these balls you see the separate components of these resting state networks. And you don't have to be able to read all these tiny script to notice that within resting state networks, you see the lines are mainly red. And between resting state networks, the lines are mainly blue. And this indicates the age effect. So within resting state networks, we find increased connectivity. And between resting state networks, we find decreased connectivity. And there were some sex effects, but these were very subtle, and most of them did not survive testing for multiple corrections. So from the means to the variances, let me first say that while we had this unique opportunity to find different genes at different ages, this is in the end not what we found. What we found that we could extract reliable component of functional connectivity, which was the same for age 18 and age 13. So that would mean a stable genetic effect throughout adolescence. And then when you split that up into genetic effects and common environmental effects, you see in green the genetic effects here, so that we could show that in about 1/3 of the connections, with heritability estimate up to 50%. And then there's some yellow, and that means common environmental effects, but those are lower in number and also explain a lower proportion of the variance. So to summarize, we found increases in functional connectivity within resting state networks and decreases between the resting state networks, and that nicely confirms what has been shown before in the cross-sectional studies. Additionally, we found stable genetic influence on cortical functional networks over time in adolescents. And finally, this reliability model enables us to remove much of the error variance or the noise variance that is present in the data. And we think that this method might also be useful if you do not have this complex design of longitudinal data or family data. We guess it would also work if you do a cross-sectional study in unrelated individuals. So please wait for our paper about that. And I'd like to thank my collaborators, and specifically Hjalmar, because he really did all of this work. And I would like to thank you for your attention.